suggesting that it might be capped with carbonate, that what we're seeing here
Lasers,
is
come on.
some effect of that secondary volcanism and tossing down stuff from the cone that could be carbonate, or it could be pillows that are descending down that slope?
Yeah, probably not so much like a whole bunch of carbonate pallets, but
good size,
the
at
depth
least.
of the feature is
Science
than
likes
what
it.
we've been on recently, so 1,500 meters, that may have an effect of what we're looking at. So I think you do have some mixed mode geology here, morphology samples.
Yeah, so John, I was noticing how round these rocks were, and that was there was a little bit of a concern to me, of course, being on this flat terrain. We are going to just be climbing all day long till we get up to the summit, so I was thinking we would wait a little bit longer till we're absolutely sure we were into some pillow flows or some type of obvious volcanic rock area before we tried to grab a rock. But if you wanted something from here, of course, we could certainly do that.
There's a candidate under the lasers at the very
well. It looks like there's no shortage of these, and they go on for a while. So I think I'd agree to hold off for a while. I mean, you can have, you can have rounded volcanic.
Might try to see if that starboard wing can go outboard anymore for you.
Typically,
get out of your view a little bit.
Wash zone of that they get rolled around.
Starboard wing coming out.
Between land and sea and the beach.
There it
so
is.
You can get nice round like river rock actually in that environment.
Video snaps in.
Whether or not this was in that environment, and also you've got quite a few of them, and they go a long way. That kind of indicates something different.
Oh, so it looks like we have a different type of sea cucumber here. These are the types that are popularly known as sea pigs. I believe this is Amperima. This has this interesting frill around the head, and like many sea cucumbers in the deep sea, it's got a translucent body, but it also has these funny little legs, which is where the name sea pig comes from, because they kind of look like pigs of the sea, except they've got like a whole bunch of legs. But they digest organic materials off the surface, freshly laid goo from the ocean floor. You can see that doing it right now in slow motion. The sediment comes along with it, goes into the gut. You can see that because the gut's filled with sort of delectable ocean goodness where it will accumulate. You can see the lower intestine sort of through the body wall towards the rear there, and eventually this one might even swim. They will poop all of that out, leaving a clean pile of sediment that will aid bioturbation and affect the deep sea floor in what is interpreted as positive ways. So thank you. We can move on.
Yeah, and I just wanted to say thanks to John Smith for his comments on the geology. We really appreciate you calling in and providing the listeners with a little context of what we're looking at.
Spitodiatomatid on the right.
Yeah, before we leave here, lower right. That would be good. Good spot, Chris.
I got the search image.
Hi, Chris. This is Susan Gottfried at Stennis.
Hi, Susan. How are you this morning?
We are doing fine. We have Mary Waldemuth from. She's our new NCEI director. She's here in the Stennis BCC along with a few other folks, and we're watching the dive. And we just are very excited to be talking with you guys.
Oh, thank you very much.
Hi, this is Mary. Oh, now I can see you. So we're watching every move.
Thank you so much for joining us, Mary. We really appreciate it. And if you've got some questions, just fire away.
And Susan, just to note, this is Kelly Elliott. I'm on the line as well, though I'm just off camera. Go ahead, Nat.
I'm thinking of the breaker. I just wanted to know.
So you're down below? Obviously,
Right now we
below
have the
the,
wind right off of our bow,
jet
and it
on
looks
the
like it
boat,
came
right?
down a little bit. It's reading like 16,
Yeah, that's
15 to like
yes.
17 knots.
Or Kelly, do you want to explain a little bit of the setup on the Okeanos?
Sure. So right now we are conducting a remotely operated vehicle dive. We have a two-body ROV that is capable of going down to 6,000 meters. We actually had a cable issue this cruise, though, so our current maximum depth is 4,500 meters. That's just because of the cable, though. And that two-body system works in that we first have a camera platform that sits below. It's attached by a cable to the ship. And then there's another cable that comes off of the back of that camera platform and goes down and attaches to the ROV Deep Discover. So I know you have 3 feeds up in that command center. And right now you're seeing the video feed that's coming off from the Sirius camera platform. And that is showing you the primary vehicle, Deep Discover, D2 for short. And then that other camera feed that you're seeing is the view coming off of the high-definition video from Deep Discover. And Chris, can you just tell her a little bit about our dive today and what we're doing?
Yes, so we are at a seamount, a flat-top seamount, which are called geos. And this is located south of Johnston Atoll. So we're inside the Johnston Atoll unit of the Pacific Remote Islands Marine National Monument, and we're trying to explore some of their deep water resources that they have in the monument, which are very poorly known. This particular seamount, we have no name for you for this because Mashkor Malik and his mapping team finished mapping the seamount for the very first time several hours ago, and we were all scrambling to wake up early and get to look at the data and pick a dive site very quickly. Normally, if we have existing data, then we can engage everybody on shore and select the site as sort of a community effort, but it was not quite possible to do that here. And by the way, I'm going to stop on that so Mary can take a look at one of the most beautiful creatures that we find down here in a living fossil. And maybe, Chris, you can tell her a little bit about the animal we're about to zoom in on.
Go ahead, Nat.
Sure. This is a stalked crinoid. This is in the species Croisocrinus ruberimus. These are echinoderms. These are the same group of animals that sea stars are in. They filter water through the arms, and this is a very old kind of animal. There are animals like this in the fossil record all the way back to the Paleozoic, and you can find sort of the remnant pieces of these in fossils and woodcuts in the Midwest, for example. So these often are termed sort of living fossils. Animals like these were actually, some people argue, the reason why deep sea research was even started, because it was thought that the deep used to be depauperate of life, and one of these was pulled up in a trawl in the 19th century or so, and people said, hey, the deep sea's filled with archaic things, let's go look for them. And so the story goes. But a very interesting group of animals with a very interesting mode of life, with a very old history.
Yeah. So, Mary, do you have any other questions for us? We'd be delighted to answer anything you've got.
Well, so for that life that you found, how come you don't have? Why don't you see more of them, like around there? How do they reproduce? Do you know?
Oh, sure. Well, it's thought that they send out larvae. I mean, the males and the females send out gametes, and they fertilize in the water column, and so the larvae actually settle out according to different environmental cues, it's thought. And so some of them may settle out near already pre-existing adults. Others may settle out on just ideal environmental sort of conditions, nutrients or water flow or what have you. The reproduction's not very well known. We know very little about these animals on the whole, and so every one of these animals that is collected or observed even is kind of a treat for scientists who study them. So there's often a lot of interest whenever we spot some of these animals for that reason. Some of these weren't even, the colors weren't even known until some of the video like this was first taken.
Yeah, it's
starfish
really
down the bottom, 2 of them.
so it's just kind of stuck onto that rock, right?
Yes, that's actually. So these are sessile. That is, they don't move anywhere. Some of them can actually move, and it's thought that some of their movements might be dictated by predation by sea urchins, which are another kind of echinoderm. So a lot of, and one of the other things that we see are small echinoderms called feather stars. And feather stars are kind of the small mobile version of the stalked crinoids. They essentially are kind of the cup and the arms, and they just kind of pop off.
Degrees, so.
And so the modern, the sort of more commonly encountered versions of these animals can actually swim and move around on their own. So it's a very interesting sort of history of life that these animals have.
Yeah, it looks really healthy.
So, Chris, can you tell Mary a little bit about your benthic animal guide and how that has become such a big success?
Yeah, I would love to, Susan. So back in 2015, after we completed the cruises in the fall of that year, we were taking a look at all of the still images that the videographers had captured throughout that season, and we had over 4,000 really stunning images of animals and midwater creatures and all types of things. And we thought, boy, this could, if we could assemble these and consult experts and get some really decent identifications on these animals, then this sure could help a lot of researchers if we put them all together in some type of a guide, if you will, and give everybody a look at what types of animals we had imaged in the Central Pacific. So we discussed this with the OER, and we had some meetings with Susan there at Stennis, and we all came up with a plan. And so the group in my lab here were composed of 4 people here, myself, Megan Putz, Sarah Bingo, and Virginia Moriwaki. We all kind of worked together to select the best of those 4,000 images, and that actually amounted to 1,700 of them. And we put some headers on there and some depth indicators and so forth. And then we sent all this to Susan and her team down there. Charles Carlton was part of that effort, and they did their magic to actually transform this into a web resource, and went back to OER too to mount on the web. And now we are in the process of creating the second version from the 2016 cruises, and we've got, right now we've selected out 2,900 images that combine the 2 years. We had to add some regional indicators on here because now we are no longer in the Central Pacific last year. We were all the way over in the Mariana Trench and Wake and so forth. So we are talking actively with Susan and her group on what the next step is, how we can improve it, how we can make it a really awesome tool for researchers around the world.
And Chris, can you also tell Mary or put the lasers on possibly and explain what that means and how that
Go for lasers?
to describe the size?
Excuse me, could you repeat that, Susan?
The lasers, how the lasers help determine size and
Co-pilot or count to stop right now?
Okay. Not a lot of time left. Pilot.
She can see
Arvina, bridge move
big it
complete.
is.
Yeah, so
Thank you,
you're
Bridge.
actually going to get a perfect example of how we use these lasers. So the lasers are 10 centimeters apart. They're just spots of light. They're very weak lasers. They don't hurt any of the animals, but it definitely helps us understand how big the animal is. So this sea star, and Chris can talk about this in a moment, this is big. This is a magnificent sea star that we actually haven't seen very much of on this cruise, right, Chris?
Oh, yes, absolutely. But because we use so many cameras with zooms and we essentially have some difficulty establishing absolute distances from a video, the lasers give us a scale, a 10 centimeter, in this case it's a 10 centimeter distance, which gives us an idea of how big the objects that we're seeing on screen are. And so in this case, this animal is at least 10 centimeters across, and that gives us a good idea of its absolute size relative to, say, looking at it under a zoom or something like that.
So, Mary, I should point out that to my left is Matt Dornbeck. And Matt, maybe you could just tell Mary what you're doing.
Hi, Mary. Thanks for visiting Stennis. Normally that's where I work. And I'm out at sea with Chris and Chris to help them with the sample collection. So we usually grab a few rocks and a few biological samples every dive, and we bring those to the surface, and there's a lot of caretaking that has to go along with that. And since the Chris's are often narrating our dives, it helps to have someone else that can take care of the samples and also log all the data and metadata that goes along with that into Susan's database called SODA. And then all that information will get transported into archives and out to the public, and we'll have access for the scientists to the samples after the cruise. And so, yeah, I'm kind of out here doing that, along with annotations of the dive as we speak. So everything that we see, I'll try to annotate to the closest possible taxonomic level and have a real-time annotation of that.
And I would just add that the recording of the data sets and making the data and sample information available to the science community is an absolutely critical part of our operation that Susan and the team at NCI has been critical to facilitating really since we brought the ship online in 2008. Our goal, this is a dedicated exploration ship, is to go to places with little to no information and to take that first look and to collect a foundation of baseline information and make that publicly accessible so we can enable follow-on research activities, so we can provide the data to managers so they can more effectively manage resources. For example, today we're in the Johnston Atoll unit of the Pacific Remote Islands Marine National Monument, and the data from this cruise will be used to help inform deep water management plans for this particular monument. And we actually have a record of working with the NCI team to get all of the data and information from these cruises publicly accessible within 30 to 60 days, but always by about 90 days after the completion of the expedition. And that is critical in allowing us to accomplish our overall exploration objective of characterizing an area, making that data accessible so others can use it effectively or do follow-on work. And it's not just making the data accessible, I think, via the repositories. I'm sure Susan and Megan and the team on shore will show you or go over it where appropriate. But they've also developed a couple of online portals to display data in real time during the cruise and to facilitate access to those data sets in a centralized way. So you don't have to go to different locations to find all of the data from one cruise. You can instead go to, for example, the OER Digital Atlas and find direct links to all of the data from this one cruise in a centralized way.
So when you take the samples,
Co-pilot, when you get up from the seat, you left the threshold in the USBLS 19.
You put it in salt water, same kind of temperature. What do you do when you get the sample on board?
Okay, well, the D2 ROV is so well engineered. The sample boxes, when they come up on deck and they're secured and we get the signal to come down and grab the samples, the water is freezing cold for us to even get in there and grab the samples. It maintains the water temperature just in a remarkable way. We have cold water buckets. We put buckets of water in the refrigerator in the lab here to have them chilled down. So we will grab the animals from the boxes, transfer them in buckets, and then move them upstairs into the laboratory. And then at that point, for all the biologics, we already know pretty much what we collected, of course, because one of the things that Matt is doing, and also we have another person as well, Nicola Rodriguez, who's helping out a lot from the University of Hilo. But what he does is at the very moment of collection, he pushes a button and we collect environmental data, latitude, longitude, depth, collection number, what box on the ROV it was actually in, everything we need to know. And as we're bringing the samples up, Matt has already printed out the museum quality labels for each of the specimens. We bring the specimens onto a light table. We photograph every specimen with its label, and we photograph it with what we call an OGL vial, which is a special vial provided by the Ocean Genomic Legacy Program that contains special buffers. And we note the vial number, and we take a subset of that specimen, put it in the buffer, and secure it for DNA analysis. If we have a request to provide an aliquot for tissue examination, then we'll take a small piece and remove that and put it in 10% buffered formalin. But most of our specimens go into 95% ethanol after we finish photographing them. And then for the rocks, so we examine all of the rocks when they come up, and some of them, sometimes they come up with small associate animals attached to them, and sometimes they don't. And if they do, then we will pull those off. We'll first photograph them on the rock, and then we'll pull those off, and Matt will be straining to create new labels for these associates, and then we'll get all of that done, and then they will get preserved in alcohol as well, and they'll become part of the database.
Ready for the move there, Nav.
So that's the process at the moment of collection.
020.
Copy that. 020.
How do you pull it off?
30 meters sounds good.
Yeah.
Copy.
With your hand?
Yeah, so most...
So a rock comes up, there's something like that starfish or something, I don't know the terms, and I apologize, comes up and, well, so you're telling me there's like a little container in the ROV. It goes and grabs it, puts it into something. So it's got all of its, what it's used to. Right. Okay. But they're pulling it off on board, right?
That's range of 30 meters, bearing 020, speed decimal 2.
Oh yeah, they can't really do it. Okay. And so is some of this data used for, like, the medical community to see if there's, something that can cure something else?
We did collect a number of sponges in the Marianas last year that Dr. Shirley Pomponi from Harbor Branch, she's a specialist in a group of sponges called demasponges, and she also is involved in looking at sponges for pharmacological value. And she had seen a couple of sponges in a group that she calls lithistidae, which are being investigated for pharmacological properties. And we collected a couple of those because they were unusual and could be promising in that respect. But most of the animals that we get are more for taxonomic examination, and we get a lot of new species, new undescribed species. And we also try to understand the community at every site. Unfortunately, we have a little bit of a sparse community today. We got some really cool animals, but sometimes when we get down here, Mary, the whole seafloor is just covered with stuff. So you just never know. Every site's different.
That would be really cool. I was wondering if every time you do this, do you see something you hadn't seen before? Or pretty highly, yes?
Absolutely. Virtually every dive, and yesterday's dive was a perfect example of that, because we actually saw a type of nudibranch, a very deep water nudibranch that was quite large, and we collected it, and none of us had ever seen this before. And Chris Ma had actually put out the word on Twitter and did some research on the internet and was able, he thinks, to identify it as a potential genus. But we also collected an unusual sea spider down here in perfect condition, because he was on a very thin coral branch, and the pilot was able to just pluck the top of the coral off and get the sea spider in completely intact. And then we got something else. We got an extremely unusual animal that we can't even go to order. It's a group that's related to corals, but we don't even understand where it belongs, and we were able to get a specimen of that as well. So yes, we are definitely seeing new things virtually every single dive.
That's great. Now, so like, when will your day end? It's like today, when you pull it up, or does it just keep going down there?
Keeps going.
It just keeps going.
They launch
Right.
it in the morning and then pull it up about 8 to 10 hours later.
Yeah.
Oh, okay.
So
And tomorrow morning they start again. Oh, they start doing side scans and figure out where they want to go.
yeah, so Mary, basically we run 24-hour operations on the ship. There is never a moment where we're taking a break. We do things in shifts. And so Chris and I and Matt were responsible for dive summaries, dive plans, the sampling database, dealing with specimens and things like that, the rocks as well. And of course the ROV pilots, once the dive's finished, they are fully engaged in ROV maintenance and getting it ready for the next day's dive. And then our mapping team takes over the space that we're sitting in right now, and they immediately start in on mapping operations and start creating brand new seafloor imagery for us. So it's 24-hour operations here.
That's good. Thanks, wife.
And how long are you going to be out there?
We're
Like, how long have you been to sea right now?
Yeah, so we left, when did we leave? The 7th. That was a long time ago.
27-day cruise.
A 27-day cruise, so we're out here for, I think, 2 more dives, and then we're going to head back to Honolulu. It'll be about a 2.5-day transit.
Oh, great.
You have one more ROV this year?
Before the,
September.
Yeah. And then you're transiting back to the Panama Canal to work in the Gulf next year, I understand.
So were we part of looking for that rescue, that plane that went down? Were we helping that? No? Okay.
No, but Mary, we've helped find some wrecks. Last year in particular, we were able to find a shipwreck off of Wake Island. And this is all based on historic records coupled with our mapping crew being able to identify a target for us to dive on. And it turns out it was a Japanese ship that was sunk in World War II. And then we also found later on toward the end of one of the Marianas cruises a B-52 bomber that had gone down during World War II as well. And that was very unusual to find one of those planes. We were able to identify it also from some of the mapping data that our mapping team had created. And so we had a whole bunch of marine archaeologists on board who are very excited to document both of these.
It's exciting. I thank you for taking time to talk to me and share what you're doing. It's fascinating. It's been a great visit for me.
Mary, we're delighted that you could make it to the ECC and participate with us for a little while. We really appreciate it.
Thanks, Mary.
Thank you.
Thank you.
Thank you, everyone.
Yeah, thank you.
Bye-bye.
Bye.
Bye-bye.
Mind if I turn that aux light off?
I don't mind.
Okay, well, we are back to our regular dive mode here, and I see we've got some people joining in on the chat room, and we've got Tina Molodtsova from the Shirshov Institute in Moscow, who usually joins around this time, so good to see you, Tina, and the rest of you, thank you very much for staying in there. And while we've been, could we image this animal, please? While we were discussing the details of our mission with the new director of NCEI, which is what's been going on, Matt Dornbeck has been diligently annotating creatures in the Sea Scribe for you. So even though we didn't call them out except for a couple of them. Trust me that they will be in the annotations. So, Scott, if you're still on the line, this may have been what you saw a little while earlier. Can you describe this?
Certainly, Chris. I am still on the line. We are looking at an octocoral in the family Chrysogorgiidae, and this is in the genus Metallogorgia, and we certainly haven't seen many of these on this leg. I don't recall, maybe more than a week ago, but certainly not in the last few days. Earlier in the dive, I said I saw a juvenile form of this, and the way I was able to determine that, it was a different growth series, is if you look at this one, you see there's a very long stem that can be up to a meter. It's usually about a meter tall, and then all of the branches with the polyps are concentrated at the end, whereas in the juvenile stage, there are no branches at the end, and instead there are little tufts of branches that come out from along the stem. It turns out that as this gets older, those lower branches drop off, and only the ones at the top remain. So it gives us a way of giving a relative age to the colony. What's really interesting is if you zoom in on those branches, you see that there's this tangled mass of what almost looks like worms right now. Those are the arms of a brittle star, and those arms are very highly coiled.
Yeah, I also noticed that we're now reading about 18.
This is a very, very tight association.
How many kill lasers are there, Mom?
Brittle star species we have only ever seen on these Metallogorgia melanotrichos corals. And furthermore, we have never seen one of these corals without one of these brittle stars. Even the very smallest ones that we can see already have a brittle star, and the very oldest ones also have a brittle star. We have on occasion seen a colony that is dead, so all of the polyps and the tissue are gone, and the sea star is still there, and the sea star hasn't left. And so Les Watling and one of his students has written a paper on this, and the idea is that the brittle star may be protecting the coral from other things that settle on it. And it can basically unravel those arms and extend them around the branches. But there's also something going on with the tissue of the coral in that we never see anything landing on the main stem of the coral if there's tissue over it. And you can see that the color there is a slight violet purple. But you can see around where the brittle star is that it's actually quite dark. So the skeleton is kind of dark, black, iridescent a little bit, but where there's tissue covering, it's more purple. So there's probably some biochemical compound in the tissue that is also warding off other larvae of other invertebrates from settling on it. And Chris Ma, I don't know if you want to say anything more about the brittle star?
I think you got it all in there, Scott.
Okay, wide, please.
Yeah, so thank you, Scott. I think we're good for science. That was the voice.
I just want to add
Go ahead.
a fascinating thing that goes back to a question I heard Mary ask before. One of the real big mysteries is how the larvae of that brittle star find the Metallogorgia. As I said, we never see them anywhere else.
Yeah.
It's not like those brittle stars are crawling around on the rocks. They just happen to encounter it and climb up. It's a very tight association from a very young age, and we think that these corals can live many decades along with those brittle stars. So there has to be some kind of chemical cue in the water that is being released from the coral that the larvae find, but you can see that, we have one colony here in the view right now, and so it's not like they're really dense. And so that must be a very, I would imagine, difficult process for a tiny larva to find the right coral when it's a lot smaller than the size we're looking at now. One of the real mysteries that we need to try to solve.
Great, thank you, Scott. And that was the voice of Scott Franz from the University of Louisiana Lafayette, and he's joining us also with James Aubrey, who's from the University of Louisiana Lafayette as well. So thank you both for participating.
Sorry.
John Smith, we were still thinking about rocks. We haven't really made any effort yet. You want us to keep going along here, or should we consider grabbing a rock? I'm getting kind of itchy to get a rock. It begins near my foot and it starts coming up my leg whenever I have a rock itch.
I don't want to know.
I don't know, John. If that doesn't stimulate you to get on the line and stop me from talking about possible rock collections, then nothing will. I'll give him you get a couple seconds here to dial in, and that's it. But while we're waiting, there is a coral dead ahead, and you probably already saw it, didn't you, pilot?
Let's just.
You did. He was closing in on this.
Well, there's a little bit of something for everyone here. We've got geology, biology, topography.
And the rock he's sitting on is a pretty nice rock, pilot.
Double points.
That's a nice rock. I don't know if it's too big, but that's a really nice rock, pilot. So John Smith has commented in the event log, and he said, okay, he's getting that same itch. It's up to his knee already, and he wants a rock.
Now, here's the question. This rock right here with the coral on it, would you like the coral left on it, removed and put in a bio box, or just leave it on the rock and drop it in a rock box?
Well, how about if we zoom the coral first of all, and we'll start there before we make our decision.
Co-pilot, I'm gonna stop that move probably like 10 meters short.
That's what I was thinking too. Sounds perfect.
Okay.
And so I'm pretty sure I know what this coral is, and I will go ahead and forward a guess. I think this is in the family Paragorgiidae and possibly in the genus Paragorgia. It's a pretty nice shape. I can't really go farther than that on this. Scott, do you have any better opinion whether we can go further?
No, I'd already that event log, Chris. I totally agree. As we were moving in towards this span, one of the things you can see is you've got this overall sort of pinkish colony, and it looked like this white fuzz kind of around it. And that's really classic for the Paragorgia, which are commonly known as the bubblegum corals. And you can see the ends of the branches here have these swollen masses with the polyps extending from them. And when the polyps are retracted, you see all those sort of grayish structures with the tentacles. They can completely withdraw into the pink part of the tissue, and it looks like big wads of bubblegum on the end of the branches.
Copy, Bridge. Thank you.
And this also has
samples, right?
Yeah, that is correct. No samples, any boxes.
Chris has snuck out. It's lunchtime here, so we're doing a little bit of shifting.
Okay,
I guess my question, pilot, we're not absolutely sure we want to collect this. The issue is that if we collect one with a coral on it, then we certainly don't want to put the coral in the rock box because that won't be good for it. I'll put this out to the community. This could clearly be a known species. I'm not really sure. We can certainly grab a rock that doesn't have a coral on it, and that's where I'm kind of leaning right now, because I'd just soon leave this animal alone if we don't think it's really a new species. But what do you guys think? I'm opening up the discussion. I'm looking at the event log right now. Tina is advocating grab them both because you've actually got a brittle star in amongst this Paragorgia. Does this look like it's of interest to folks? And like I said,
all I can say, Chris, is this is a pretty classic-looking Paragorgia, but I don't know enough about the taxonomy based on this sort of visual that we're getting to know if this is something known or unknown. I guess if you think that the rock would fit into the bio box, then it's worth it. But if not, I would say
this outer one's pretty big.
Otherwise, we would normally just take a clipping of the colony to leave part of it there.
Yeah, and I think
pilot, let's pass on this rock and leave the coral alone because it's not clearly.
Now there are some other rocks within reach as we sit.
can we get, if we're going to look in this group, can we get something with girth, as you know, maybe 15, 20 centimeters
Like
across?
that one?
Is
I
that
don't know if I can
flat or
reach
is that
one.
girthy?
Let me...
How about the one to the right, that kind of rounded?
I see that, yeah, okay. Let me reach out and see what I can do here.
Hydraulics look good. Ready when you are.
The one with the lasers on it?
Video is changing out.
Well, he's poking around at rocks. I wonder if you grab that one there, see if it'll wiggle. Come on. It's going to move. Is it glued? Here's the problem, of course, as many of you know, you can get fooled. A lot of these rocks can be glued into the substrate with this manganese crusting. Oh, nice job, Dave. That's wonderful. That's Dave Wright, the pilot. He was able to actually unstick this one, which doesn't happen that often.
Oops. Starboard. Oh, wait.
I stayed too long on the index button.
Okay. It's loose.
It's loose.
You think it's starboard, right? Pretty big?
Yeah, starboard. It'll be fine. Starboard. It's just
okay.
Momentary.
Let me know when you want me to bring it inboard. Slide it in.
Yeah, it went into spatial correspondence on me, and I got into a tug of war there. This is a better grip here anyway.
Oh, yeah. Not going anywhere.
Okay.
I can rotate your Zeus up if you'd like.
Yes, please. Do the...
Coming up.
I don't want to sediment this coral here. Let me go over to the side here.
Holding there. Rotator's at -12. I can come up more if you want.
That's all right. Go to Zoom video. We got Tara's in. Tara! Hi there. Okay. Little less zoom. Just a smidge. That's good. Thank you.
Sizable geo collection there.
Looks pretty clean. Not a whole lot of other things, but it looks like a really good rock.
Oh.
In fact, it looks like a terrific rock.
The adoration of rock. Wide, please, video.
I'm going to bring your starboard wing inboard.
Wing in, please.
Coming in.
There has got to be a basalt center in the middle of that thing. Famous last words, but we certainly hope so. We are trying to get something that has basalt because basalt is absolutely crucial to sample on these seamounts so that we can get an idea of their geologic age and geochemistry. All of these rocks will be shipped to the Oregon State University repository that's run by Dr. Anthony Coppers and his staff, Maisie at Chesapeake.
I agree.
And that will happen beautiful, a perfect fit.
Eagle has landed. Going outboard with a wing.
So, thank you very much, pilot. That's terrific.
Yeah,
so this manipulator is a force feedback type, and I could actually feel the weight of that rock there.
I have to adjust your Z
So
bias now.
John Smith, his voice we heard in between there, he also concurs. That was a good rock, so thank you very much. While Dave was collecting that rock, Chris Ritter, who's our ROV ops director here, he came in and whispered in my ear and told me that the weather has actually settled down, and right now at least we're good to go to continue the dive. So this is very, very good news. He's been monitoring it every 30 minutes, and I'm just passing this along to our participants.
Looks like you're done with hydraulics.
Yep.
Everything's stowed, so I'm going to
That is good news. And Chris, let me say, well, we're just seeing the last of that Paragorgia with the brittle star. Since I talked at length about the close relationship of the brittle star with the Metallogorgia, I'll note that Tim Shank and his group worked with some Paragorgia and the brittle stars in the Paragorgia branches from seamounts in the North Atlantic. And I believe he found about 60% of the time you always saw this one species of brittle star associated
Do you
with
want
the
to keep moving, co-pilot?
or one other species, Flexor.
Yeah, let's call it a 30, 20, 020.
It was restricted to just a couple of different kinds of coral.
The fish, I think.
Just how they, excuse me, there's a fish to the left, or right, excuse me. I guess it's a fish. But yeah, how they do that, this is just a mystery, Scott, how they coordinate like that and how they find each other. Okay, I'm going to let our fish guy, Bruce Mundy at the NOAA Exploration Command Center, take over.
Okay to zoom.
Bruce, let us know what you see here.
Okay, this is a different type of fish than we've seen earlier, except for the one that was hiding underneath the rock or in a little lava tube. This is a cusk eel in the family Ophidiidae. I'm looking at the fins on the side to see if they're in sort of two parts, and it looks like it's only one part. This one has a rounded head and a rather small eye, but it's in the genus Basozetus, and there are several species in that genus. I can tell that it's a cusk eel because of the eel-like body. The dorsal fin on the top and the anal fin on the bottom are joined with the caudal fin at the end of the tail. And then it has these little whisker-like fins up near the throat, reduced pelvic fins on the body. So the Basozetus always seem to be up off the bottom, swimming around, not immediately down next to the bottom. And they probably pick out small crustaceans and other animals, maybe even gelatinous zooplankers in the water, drifting past over the bottom. So a cusk eel in the genus Basozetus.
Thank you very much, Bruce. And that fish was swimming in a very good direction to accommodate Scott France there in the University of Louisiana, because we have a very tall bamboo coral right in front of us now. So not as tall as, what was it, yesterday or 2 days ago, but still an impressive specimen.
Yeah, I'm glad we're getting to see this one. We didn't get a close-up on the one that we saw when you were talking with Mary, and I suspect that this will be a different species from the smaller unbranched coral that we had a close look on early on, and I suggested might be an early stage of the trident candelabra. I think this is going to be something quite different, and the way we would determine that is to look closely at the polyps. And I can already see from a distance these look like little volcano-shaped mounds. And so I would suspect that's in the D2 clade. And Chris, you were noting earlier that for your group who, after these cruises, works on annotating all these video and to the best of your ability, making good note of the number of different species we're seeing, this becomes quite a challenge when you don't get a good close-up.
Yeah, but Scott, this has got some distinctive polyps. They look like volcano polyps to me. Would you agree?
Yeah, that's what I just said.
Oh,
you can already
sorry.
see these had volcano polyps, so I think this is D2 clade. Perhaps the genus Ecnomyces, which is characterized in part by polyps of that shape, although there may be some other taxa that have that shape. What you can see here is the polyps have all withdrawn their tentacles, and so the tentacles have been kind of folded down, almost curled inside of the mouth, and that's what gives them that distinctive shape.
Yeah, thank you.
Have a little more
disturbed by something.
Okay, let me come down a little.
Thank you, Scott.
Okay.
Yeah, sorry I missed that. I didn't hear that volcano polyps, and of course we're multitasking here. Again, trying to keep up with annotations and everything else. But yeah, this is a very distinctive looking one.
I suspect when I was chasing the fish, my prop wash alerted this to my presence.
Quite possible, but we're glad it did, if that's the case, Dave, because he'll open up when we leave, and this gave us a look at how the polyps close up, which is very helpful. So I think we're good for science whenever video is done.
Coming away.
Yeah, that was a great image. Thank you.
All right, pilot, you're off to my starboard. I did turn a little bit off of 020, so.
I'm just going to pull my tether away from this before I...
Yeah, good call. It's blowing to starboard right now, the tether.
Get back to where I belong here.
I'll have to turn as like 4 degrees. I'll be back on 020. We're about halfway through the move, looks like.
Yep.
Or no more than that.
Yeah, we only have probably like 8 meters, 10 meters left.
This is NOAA ship Okeanos Explorer.
Thanks.
We are on a seamount that had never been mapped until earlier this morning.
Should get
and we're diving
steeper and
on
steeper as we
a
get
small
near the top.
and what appears to be a volcanic cone on the top of a flat summit.
We are 550 meters away from the top.
The seamounts are called guyot. This cone provides a couple of opportunities for us, which is why we decided to target it for the dive today. One is that we can get some possibly very good rocks for dating it, and secondly, we often find corals and sponges and other animals taking advantage of the topography that's rising up off of the summit. So, pilot, we're probably good on this. We've got a bunch of images of these already. Can see if we can find another incredible nudibranch or something weird like we did yesterday. Or even a weird coral. We saw a weird coral a number of dives ago that is now tucked to bed in heat seal bags, and in our cooler chest here he's been changed out and babied and everything else, and now he's
we're done with that move.
Ready to get back to Honolulu
Couple good
for the
heaves
end
in
of the
there.
cruise, and then he'll get shipped off to the Smithsonian.
Bridge, move complete.
So, co-pilot.
Copy, bridge.
Co-pilot, engage the weird filter on the sonar, please. Let's find something weird.
One of these pro-isocrinus ruberoinus.
Chris Paul or the samples?
I don't know what the weird filter
What's that? Excuse me?
I was asking if you were talking about Chris Ma or the
Nav's in
samples.
control. I don't have a mouse for this. Actually, this mouse might do it.
Funny you should mention Chris, because he just arrived. And I'm going to switch with him and go to lunch and let him respond to that. I was talking about the fact that we tuck stuff to bed, and I was talking about samples, but he was wondering whether I could have been talking to you too, about you too, because he hadn't heard your voice for a while.
Hi, Scott. How are you?
Hi, Chris. I just heard the other Chris just say that somebody was looking forward to getting back to Honolulu and being shipped off to the Smithsonian, so I thought maybe he was talking about you. You must have talked about samples, surely.
Yes, certainly we're at the Smithsonian, which is the depository for specimens collected on this expedition. We're looking forward to having them and making them available to the scientific community. Also, don't call me Shirley.
Nice reference. Nice one.
Where did it go? It's hiding. It's hiding right there.
So, we're back, and having been inspired by our galley's fine pastry and dessert chefs,
Okay, wide, please.
I'm now looking for cookie stars. Hopefully a
What?
goniasterid will turn up.
Give a poke ahead here, Levi, and we'll swap.
Okay.
Co-pilot 020 still looking good for the slope.
I think so. What does HiPAC have?
So HiPAC agrees with that. It's about 010 on HiPAC. So if you want, we can keep moving. Co-pilot.
Okay. Carry on.
Yeah.
Copy.
Video, let's snap.
Sure thing.
Center. Show what this little guy is.
Oh, good eye.
Oh,
So it's going to be 020 30 meters.
black
Okay,
coral.
co-pilot.
Roger that.
Watch this. You get a closer look at this. I just kind of.
If we could, please.
Yeah. All right. Yeah, video. I'll get
Great.
closer. Yeah.
So
Go ahead, Matt.
I'm not sure if we've seen one of these in the time since I left, but this is an Antipatherian, also called a black coral.
Speed decimal 2 coming up.
And I believe...
Okay, video command.
Our shoreside expert, Scott France, may know a little bit more about these, or maybe Tina Molotova.
Okay. Partial.
Yeah, Chris, I'm still just waiting for the
you got it?
I can get a better look at
Ah,
it.
very good.
Hold that.
Well, you're absolutely right that it is a black coral, and so it is somewhat distantly related to the other octocorals that we've been seeing before. In fact, this would be more closely related to the anemones than it is to the octocorals. They've got polyps that have exactly 6 tentacles associated with them, and when we do these close-ups, one of the things that you'll see that are different between black corals and octocorals that you can get used to, and so you can identify them yourself. I'm talking, of course, to the general public watching, is that the tentacles of the black coral do not have little side branches, which are called pinnules. So this one, given its branching structure, may be a very young Bathypathes.
There's something.
There's a
Zoom out a little bit.
Slight possibility that it could be Alternipathes, although I don't really think so. You mentioned Tina Molotova from the Shirshov Institute in Moscow. She is certainly more of an expert than I am, and so.
Hey Scott, we've got something really interesting here. There's a little purple soft coral that's on the rock. I mean, it doesn't look like Victorgorgia. It looks like literally two polyps on the rock that are purple. It looks like
so there is
octocoral.
An octocoral that grows kind of like a mat.
I assume that's Mac's Zoom video. It's a small subject. Yeah, it's a little guy.
It's either overgrowing corals or rock or sponges, and it's called Trachyphila, or I don't remember the other genus name. I think it's been transferred one to the other. I'll have to check that out.
Is that stoloniferous?
Possible. It is, or it's just such a small Victorgorgia that the color is throwing us a little bit.
Neat. Okay, but that was the Max Zoom, right?
Yeah, that was Max Zoom.
No worries. We're good.
That's what we got.
Yeah, we'll keep going.
You can see that the tentacles here are pinnulated, so it's a really nice comparison to the black coral. So right away we know this is an octocoral. Also, that term octo has 8 tentacles. The black coral is a member of the hexacorallia tentacles.
Line back up.
Thank you.
You guys called the move in at 020 or?
Yep, we
Tina just reminded me
about
Clavularia,
probably halfway through the move right now.
the other genus compared to Trachyphila.
Thank you.
See something on this rock.
Thank you.
Video that's snap on. Is it possible?
I see.
Urchin, maybe?
Is it an urchin?
Yeah.
Ooh, I love urchins. Ah,
partial.
We saw one of these earlier. It might be a little different, actually. Interesting. But yeah, we can zoom in if we've got a moment. Zoom. Oh yeah. So this is, I believe. That's curious. It seems to be a relatively small aspidodiadematid urchin. The spines are a lot shorter than the ones I'm used to seeing. Usually they bow out away from the body, and we saw one earlier today, just before we began sort of the interactive session. What's interesting is that top structure there. Oh, it's withdrawing its anal sac. That top structure there is what's referred to as an anal sac. It's an extrusion from the intestine that opens up and is where the poop comes out. So they eat. The mouth is actually on the bottom, and the intestine goes up through the central sort of body, and then it's extruded out through the top. And we actually saw one with fecal pellets in the sac in the earlier one that we saw. But it looks like it's withdrawn. Perhaps they do so when they're finished, or when they're. This is in the Aspidodiadematidae. As I had mentioned earlier, usually they're crawling around with really extended spines. So it looks like but this one also has tube feet in play. So
Okay, video is clear.
Yeah, we can move on. Thank you.
Got the ship moving, so I gotta kind of keep moving here.
No twists in the tether. No hint of.
No, nothing fishy going on there.
No hint of pigginess.
Yeah, it looked good to me too.